And now we come to source localization. And here we have a fundamentally ill-posed problem. By ill-posed, I mean that there are an infinite number of solutions to this problem. Right? So given that we have data from outside the head that we feel came from inside the head, so ignoring the possibility of noise, we have to figure out from our sensors, and again, a very limited number of sensors, even with MEG, if we say we have 106 locations, multiple sensors at each one, but still 106 locations, that's orders of magnitude less than the number of neurons or even the number of distinct brain areas. So if we take sort of 180 per hemisphere as an estimate of the number of distinct brain areas in terms of function and structure, then 360. So our MEG sensors is sort of 1/3 of that. So each sensor is, by definition, picking up activity from multiple brain areas if they're involved. EEG, it's worse because we have fewer electrodes, and then you've got all the volume conduction. So given this relatively limited sampling of the data, we have to figure out how many sources there are in the head, where they are, how strong each one is, and how its strength varies over time. And when I say locations, it's both location and the orientation of that dipole inside the head. So that's a lot to figure out, and basically there's an infinite number of distinct combinations of those four things that could generate exactly the same pattern of activity at the scalp. So it makes the problem hard. This slide has very poor contrast. That's unfortunate. It's from the book, and basically it's showing that given 4 different dipole locations and orientations, you get the same scalp distribution of effects. So it's really just visually describing the inverse problem that I just described. You can't see it here. You can always look at it in the book or just take my word for it. On the other hand, so that's the inverse problem, or you could say the inverse problem is trying to solve this. Inverse solution would be how do you actually solve that. But generally we talk about the inverse problem and then forward solutions. So a forward solution is if we know the number of sources, where they are, how they're oriented, how strong they are, and how they're changing in activation over time, then it's very trivial to predict what that's going to look like at the scalp. And you can do that with more or less complex head models. I'll come to head models in a minute. So depending on how you actually model the head and the brain and that sort of thing, you can get some variation in output. But in principle, this is a fairly tractable problem. And given a set of values for these 4 variables, there's one solution, basically, given assumptions about the shape of the head and all that other stuff. So there's unique solutions. So it's not ill-posed at all. It's very well posed from a mathematical perspective. And this is the only hope that we have in solving the inverse problem, is if we can come up with a forward model that makes sense and is plausible and matches our data. And that's really how source localization algorithms go about solving the inverse problem. So comparing EEG and MEG in terms of source localization, because all the methods I'm going to talk about are equally applicable to both EEG and MEG. They're not necessarily as effective, but they can be used for both of them. So the issues with EEG are that we have volume conduction, so there's a bigger range of possible solutions that are feasible given what we know about the physics of how that signal is propagated. And we typically have fewer sensors, so 64, maybe 128, maximum 256. And when you have 256, a lot of those end up being like on the face and on the neck and that sort of thing. So in principle, you have more comprehensive coverage to capture the other parts of those dipoles and so on. But you're also going to get a lot more noise just because those are sitting right over muscles and so on. So the quality of the data is not necessarily as high. So you have fewer sensors and more possible solutions with EEG compared to MEG, where the possible origins are more constrained by the fact that these magnetic fields drop off so sharply with distance, the fact that you have 306 sensors, and 3 different types of sensors, at least in the electroscanners, that have differential properties to these magnetic signals that help you better sort of triangulate how the fields are distributed. And that's why MEG is so much better at source localization, is because of these points here. And so in my humble opinion, but certainly not only my opinion, MEG source localization is at least believable. It's not always right, and we'll talk about that, but it's at least believable. Whereas EEG source localization, I think, is largely voodoo, and I would put very little stock in most, even published studies of EEG source localization. And I think the issue there around publication is that mathematically there's nothing wrong with the algorithms. As I said, the same algorithms can be used for MEG and EEG. And so if you take some data and you run it through one of these validated algorithms and you do things the way it's supposed to be done, you're going to get some answer. And you're within your rights to believe that answer and put it in paper. And assuming the peer reviewers understand source localization and are able to critically evaluate the methods, there's no reason not to publish that. But the amount of faith or belief you should put in those results should be quite weak, which is something that I think a lot of the time isn't the case. I think people either don't fully understand or just want to believe their source localization results with EEG and do put a lot of stock in it. The qualifier there, major qualifier, is that for certain things where, especially if we're talking about like an early sensory response, so an early auditory response or a visual response, somatosensory, motor activity, we know a priori where those things should localize. And so on the one hand, it's sort of, why would you bother localizing if you already know where the signal's coming from? I'll come to that in a minute. But at least in that case, if I do source localization on auditory evoked potentials, like an auditory P1, and my result tells me it's in the bilateral superior temporal lobes, I'm going to be pretty confident in believing that, because that aligns with everything we know about the brain and the organization of this sort of thing, right? If it puts the result somewhere else, that's when I should start being quite skeptical. And why you might do that, assuming it goes to the right place, there are reasons to do that. One is that it's a way of trying to, remember when I talked about EEG, I talked about latent components, and the fact that what we see as waveform peaks are the combination of all these different underlying sources of activity. So if we can isolate from all that combined activity using source localization, we can say this part of the activity is coming from the temporal lobes and not other aspects of that waveform that we're seeing. It's a way of actually finding those latent components in the overall scalp recorded components. So source localization can be a way of sort of parsing apart your data and isolating the time course of activity from distinct brain areas to the extent that it is believable and so on. And that's the big use. Another one I saw that was kind of a cool study, and again, it was not novel in the sense that we already know that the auditory cortex has what's called tonotopic organization, meaning there are parts of the auditory cortex where there are neurons that are tuned to different pitches, so different frequencies of sound, and they're arranged in a logical sort of order, so that within a patch that's tonotopically organized, one side will be most responsive to low frequencies, low pitch tones, and the other end will be most responsive to the higher pitch tones. And so using source localization, a research group was able to show this by presenting auditory stimuli of different pitches, recording the auditory evoked potentials, source localizing those, and showing that the localization actually sort of progressed along the temporal cortex in a way that was consistent with tonotopy. We already knew tonotopy existed, so it wasn't like they proved that, but it was a nice demonstration that it was possible. It gives you some faith that in certain contexts, EEG source localization is believable, because it's sort of showing you what you would expect to see. And potentially, if you're interested in studying tonotopy in people in some way, see if auditory training or hearing loss or something affects tonotopy, it tells you that auditory evoked potentials might be a low-cost way of doing that rather than having to do MEG or fMRI or some more expensive technique. So there are use cases for source localization with EEG, but I think they're relatively limited, and I think that's the suitable amount of skepticism isn't always observed in the published literature around that. Okay, so moving on to the actual approaches to source localization. The most venerable and simple is, I should really say Dipole's up there, because BESA is just the software that sort of was first developed for doing dipole localization and still widely used for that purpose. Incidentally, all the figures that I show with like the little red pointy dipoles and the projected fields were generated by BESA, or actually a free part of BESA called BESA Simulator that you can download and just model dipoles and stuff. In this case, what they're doing is the BESA software allows you to specify how many dipoles you think there are and where you think they are. And based on that, it's going to calculate the forward solution from all of those dipoles. So, and you have to have some idea around the time course as well. So it's going to generate a forward solution from those dipoles. And the critical thing is you give it your data, you give it your set of dipoles and how, where you think they are, and it's going to try and figure out the optimal time course of activation in each of those dipoles to produce the observed ERP activity that you recorded. And so in this case, where you see source waveforms, we have 7 different source waveforms. Those correspond to the 7 dipoles that were modeled here, and you can see where those are. So again, this is making that point that I showed before, that it's a way of pulling out the latent components. What I didn't show here is the original ERP waveform, but it would basically be sort of the summation of all of those together. So it's taken something that might have only one peak at a given point in time and showing that actually, that peak is being contributed to by, if we look at the latest peak in here, it's being contributed to by multiple different sources in the brain, whereas the earlier peak is only being contributed to by maybe sources 1, 2, and 4. So it does help you sort of parse apart the time course data that way. This is up at the top is just the scalp maps of the overall activity. The other thing BESA will do, if you let it, is you can pre-specify number of dipoles and locations, but you can actually allow it to adjust the location and orientation of the dipoles. So, you might broadly think, oh, this is a visual task, as this one was. You can see the dipoles are largely kind of pushed to occipital lobe and inferior temporal lobe, typical visual areas. So you can have predictions about generally where these are going to be, but obviously from person to person, the actual locations are going to vary a bit. And so you can say, okay, I think these dipoles are going to be in these locations, but you can define a sphere of, say, 2 centimeters around each dipole, and BESA will sort of change the location and the orientation in ways that optimize the fit between what it's predicting from its forward solution and what you actually have as data. The issue with, well, I'll come to the issue in a sec. This is just another example, and really the only, well, 2 things here. So one is that it's showing for visual evoked potentials dipoles for the C1 effect, which is like the earliest visual response before the P1, and then also for the P1 effect and the P1 attention effect, meaning that the P1 was modulated in its size depending on whether people were paying attention to the stimulus or not. So you're getting 2 dipoles for the C1 and 4 dipoles for the P1. And then they're superimposed on the actual MRI from an individual so that you can see, rather than just sort of inferring locations from these line drawings of the head and the brain, you can actually see relative to an anatomical MRI where they are. For better or for worse, it obviously helps you visualize where they are, but it might create a false sense of belief in the level of accuracy of these techniques, for better or for worse. Again, the issue with dipole modeling, well, one issue is that every other imaging technique we have, say fMRI in particular, because fMRI has quite reliable, you don't even call it source localization in MRI or in fMRI. Like we know without doubt when we see something in a certain area of the brain in fMRI, that's where that signal came from. There's no sort of mojo around getting that out. So if we take MRI as kind of a gold standard in terms of what spatially distributed patterns of activation look like in the brain, they don't look like little dots. They look like blobs, right? If you've ever read an fMRI paper, you see like blobs of colored activity in different brain areas. So we know that neural activity tends to be distributed over the cortical surface rather than highly focal. So the dipole model is a mathematically convenient simplification of what brain activity actually looks like. But it does mean that you're trying to represent something that's distributed by a single point, and that makes it easier to do the math, but it's also likely to make the math be wrong. And so these don't necessarily reflect the full extent, and sometimes the dipoles can sort of be biased by, they're not even necessarily in the center of a blob of activity, but sort of biased in one direction or the other just by the shape of that blob. So that's, they're not the most accurate model of true brain activity. The other is that although we're using a forward solution and trying to come up with a forward solution that best fits the data, that's very sensitive to noise in the data. So you want to make sure you really clean up all the artifacts and that sort of thing. But even still, some EEG data, as you'll no doubt experience, is quite noisy. And the noise really limits your ability to do source localization because the assumption of the models is that all of the information, all of the variance in the data is informative, and it should try and fit that. So having noise can actually cause dipoles to be completely wrong, just because they're trying to fit to the noise rather than to the data. Another issue, there was a study that was published quite some years ago where they took, it was like 8 or 9 individuals ranging from undergraduate research assistants in a lab to faculty members who had experience with EEG and source localization, and generated some data where they used modeling software so they knew where the sources were and how many there were, and generated the predicted pattern of ERPs based on that, added some noise to it. Without telling anybody how many dipoles or where they were, they gave this data to these 8 people with various levels of experience and training and told them to run BESA and come up with a dipole solution. Every person generated a different result. And the people who were more experienced didn't actually generate results that were closer to the known ground truth than the people who had less experience. And so that should make us all very suspicious of the whole process of dipole localization. Now, again, that's not the situation where, like here, we've got visual evoked potentials, C1, P1. These are early effects. We know from other things like fMRI and direct recordings from the brain in neurosurgical patients roughly where these things are generated, and that they're all generated in the visual cortex. And then we localize them, and the dipoles are put in those locations. So we have converging evidence that gives us faith in this. Whereas in the model case, it was made-up data, and it wasn't necessarily biologically reflective. It was a lot of distributed sources and so on. So this is more of a best-case scenario in the slide, whereas that study was kind of a worst-case scenario. But in reality, in most studies, especially if you're dealing with higher-order cognitive ERP components, you don't know what the ground truth is. You know, we have from fMRI studies, we know roughly where things like the P3 and the N400 might be generated. But we also know that there's a big disconnect between fMRI and ERP data in terms of what they're measuring. So just because you give somebody an N400 task and you see a distributed pattern of activation with the fMRI, that doesn't mean those areas are all contributing to the ERP effect, because maybe some of them are closed fields or whatever, right? So there's a lot of uncertainty, and in real brain imaging data, we rarely know what the ground truth is, which makes it hard to evaluate how good your source localization results are. And so it comes down to a combination of, you know, intelligence and common sense, and looking at the literature, and the reliability of the algorithm itself, and having algorithms that are less dependent on subjectivity. So dipole modeling is the most dependent on the subjective sort of user input. As we go into the other ones, which are all basically broadly called distributed source models, one of the things that's very important, applies to dipole modeling as well, is modeling the conductivity of different tissues. So we know that skull is a poor conductor of electricity. We know that skin is relatively good, CSF is good, brain is pretty good as well. And so we at least want to model the electrical conductivity of these things, because we're going to model things that are inside the head, and then we want to model how those propagate out to our electrodes outside the head. The simplest models use constant values for each of those, so you can just assume that the skull has a fixed level of conductivity, and obviously it's, or maybe not obviously, but it's easy to measure that, because you can take a cadaver skull and just put electrodes on either side of it and measure how much attenuation of the current you get, right? So that's pretty easy, same with skin or any other tissue type. Depending on the level of resolution that you want, you can also start modeling like skull versus softer bones in the head, fat, the eyes have different conductivity, your cerebellum has different conductivity than your cortex, muscle. So there's a lot of additional information that you could factor into this, which can, you know, in turn increase the level of complexity of the math involved and computational time and demands, to the point where the most high-end source localization algorithms require like massive parallel supercomputers to compute. They're not something that people are doing on desktop computers. The other thing is that in reality, even though we can measure or, you know, provide a conductivity value for the skull, in reality the skull thickness within an individual varies across the skull, and between individuals that thickness varies as well. It's a function of age and just individual differences. And so you can sort of approximate a one-size-fits-all, but again, the more advanced ones would actually use scans of the brain and estimate the thickness of the skull and adjust the conductivity values depending on the surface of the skull. So there's a lot that can go into that. The reality is a lot of head models in source localization algorithms just assume that the brain is a sphere, that the head is a sphere and the brain is a smaller sphere within the head. And they do that because the math is way easier for spheres than it is for complex brain-shaped sorts of things. This has changed over time as computers get stronger and algorithms get developed and built on and improved and that sort of thing. And for some things like dipole modeling, it turns out that the choice of head shape has a relatively small impact on the results. They're more like just changing the amount of noise in the data might change the results more than the shape of the head. But we can do a couple of different types of modeling of the head. The finite element model is one where you're modeling the surface of each tissue. And so what you're seeing here is different colors. So if I get this right, I think the red is the yellow is the skull, the red is the CSF, the green is the actual cortical surface, and then the dots are points on that cortical surface. So you can take something like this structural MRI scan, and there are algorithms that are pretty accurate at just automatically characterizing these different tissue types based on sort of a template of what the brain normally looks like and the intensity of the images, because all these different things. The skull is quite dark on these images, whereas and so is CSF. Fatty things are more bright, so it's fairly easy to identify these things. And then once you've characterized where the cortical surface is, you can do something called tessellation, which you see the start up there by modeling dots all over that. Tessellation is basically sort of connecting, putting dots all over the head and connecting them by triangles. And then the triangles just vary in their size. And from a mathematical point of view, what this means is that now we have a way of representing the skull, or the cortex, in a numerical way. So we get a value at each one of those vertices, so each point on the triangles. That's a lot less detailed than if we sort of represented each pixel in that image. So there's a lot less data, but it's still fine-grained enough that it's sufficiently resolute for our source localization purposes. And the other thing about that is it actually, although less relevant to do in source localization, you can do things like then take that and flatten out the map of the brain so that you can see, and that's actually in like the previous one, that bottom right picture is a flattened cortex. So the sulci are the dark gray and the gyri are the light gray. And so that from a visualization perspective, that makes it a lot easier to see, because when it's the sort of normal folded cortex, if there's activation down inside a sulcus, you might not be able to see that from the exterior view. So anyway, you can basically create a model of the surface of the cortex. And where this is valuable is not just in that we want to model the conductivity of different tissue types separately, but because modeling the cortex and the actual surface of the cortex gives us huge constraints on the source localization, right? Because now we're not just sort of saying the signals are coming from somewhere inside the skull. We're saying we know that our signal is being generated primarily by pyramidal neurons that are aligned, who have a particular orientation perpendicular to the cortical surface, and they're located in the cortex. So we can exclude all the white matter, the ventricles, non-brain space inside the skull from the space of possible solutions that we might consider as valid source localization solutions. So that provides huge constraints and makes the inverse problem far less ill-posed, because now there aren't an infinite number of solutions. We've already taken a huge set of those solutions and thrown them out. And that makes things a lot easier. That actually just twigged me to something. Has anybody watched the Imitation Game movie? So it's about cracking the Enigma code from the Germans. And so they, it's about Turing building the first computer, really, and trying to use it to crack the code. And they go for a long time. And the big problem is they have to iterate through so many possible solutions, and the computer's not fast enough to do that before the Germans switch what the code is the next day. And then they have this insight that if they can constrain the search space and exclude things that are completely infeasible solutions, they can reduce the number of things they have to test and go through it more quickly. And that was basically how they cracked the code, is they just figured out, there were certain things like Heil Hitler was in every message that the Germans were sending. And so they just figured out, okay, if this thing means H and this thing means E and so on, then we have part of the code already cracked, and that constrains our source space. So it's the same sort of concept as source localization, is you're throwing out a lot of solutions that are just infeasible, and then you can have a limited number that you have to test, so things go faster, and you have more faith in the results of the solution. So anyway, with these distributed source models, so the difference between these and dipoles is they're actually doing essentially the same thing. The distributed source model is putting a dipole at each point in the head. So rather than constraining yourself to 4 dipoles or 7 dipoles, or having to make some a priori prediction about the number of dipoles, you just say the whole cortex is dipoles. And we know that there's neurons in all these, all across the cortex, and they're all going to be generating some level of electrical activity. So let's model what combination of activity levels at all of these dipoles best matches what we see from the outside of the head. So it's dipole modeling. It's just dipole modeling on steroids and with some intelligent constraints, right? So not only do you know that the dipoles are located just on the cortical surface, but you also know that they should be tangential to the cortical surface. So if you know how the cortex is folded at each location, you know which way that dipole is pointing. So there's no ambiguity or guesswork around how that dipole is oriented. So it's more biologically plausible and more sort of constrained than the typical dipole modeling. And so you do that, and you project to the cortical surface, and just go through this iteration by iteration, trying different combinations of weightings of strength of these different dipoles until you get something that is the best fit to your observed data. So it's still computationally quite intensive, but at least you have a lot more constraints. And then there are other constraints because these models will have certain biases mathematically to produce certain patterns of results. So people sort of studied that and tried to undo those. So you can get something like this. Typically what you see is like the one on the left, where you've got the peak level of activity is in the auditory cortex. This was an auditory stimulation study. And then some distributed activity around the superior temporal lobe, below that in the superior temporal sulcus, above that on sort of the lower bank of the parietal lobe there. And what's interesting and gives you even more faith in this is that although these look like sort of weirdly shaped blobs, if you look like this one in the superior temporal sulcus, the lower one really respects the sulcal boundaries, and it doesn't cut into, like, there's a gap here in the gyrus. So it seems to be respecting inherent sort of aspects of cortical anatomy that we know are real, and sort of matches how the brain is organized. So even though it's sort of a distributed blob compared to like the little point that you get for a dipole, it makes sense biologically. The one on the right there is showing the results of the same source localization, but rather than just color coding the intensity of activation, it's using arrows where the orientation reflects the orientation of the dipole on the cortical surface. It's a little messier, but it's another way of visualizing. So within the class of distributed source models, there's lots of different algorithms. So there's one called minimum norm estimation, or MNE. Incidentally, the software we use for EEG analysis was first developed for MEG analysis, and it's called MNE Python. It's a little confusing because that's just they developed software to do the MNE source localization, and then sort of built that software out to do everything else and use EEG as well, and so on. So, but MNE is one algorithm, dynamic SPM, DSPM is another, Loretta or S-Loretta are other ones. So those are common terms that you'll see if you read source localization papers. They're all based on similar principles, and it's just the sort of mathematical details that really differ between them in terms of how they apply those constraints and weight them and determine what's the best result and so on. The discomforting thing about them is that they don't all give you the same result. Hopefully they'll give you similar results. If you have cleaner data, you're more likely to get conversion results. If you have noisier data, they're more sensitive to noise in different ways, so you can get more divergent results. But that's, at the end of the day, the biggest issue with MEG is that although you get these sort of fMRI-looking source localized results, those results are still dependent on the algorithm that you used and the quality of your data. So again, where fMRI, the location information is quite accurate, and there's no sort of question or magic around that. With MEG, you have to take these. It's far better than EEG, but you still have to take the results with a grain of salt. And sort of interpret them as evidence in favor of or against some hypothesis, rather than being definitively this is where it's coming from. So the last technique is beamforming, which is still a spatially distributed source model, but it's taking a very different approach from those other ones. And the difference is that rather than simply modeling, doing the forward solution, it's also applying a spatial filter. And what this means is you compute the forward solutions for each point on the surface of the cortex, or even you can do it volumetrically with every point in the brain if you want to. And then you get the activity for one location in the brain by subtracting out the effects of the dipoles from every other location. So it's like an additional step, essentially, at least conceptually, where you say, okay, I want to zoom in on this area. I have a forward solution from that area, but it might be contaminated by other information. So let's first say, okay, what in the data can we explain by the activity of all the other parts of the brain? Once we've explained that, what's left that we can associate with this one particular part of the brain? So in principle, it's the most principled and best. In practice, again, the issue with all this is that in practice with real brain data, we typically don't know the ground truth. Unless we actually had physical electrodes stuck in the brain so we knew exactly where they were, which is not feasible, we can't know the ground truth. But in principle, the beamforming makes the most sense and has the most constraints. One of its biggest limitations, such as it is, because it turns out not to be as big a limitation as originally thought, is that this assumes no correlation between activity at each location in the cortex. Right. So if you imagine that in one case you can imagine two parts of the cortex that are fairly close together, part of the same active brain area, obviously they're going to have highly correlated signals. But also you could have two different brain areas where one's sending information to the other, in which case the activity is going to be highly correlated just because the activity in one is being driven by the other, so they should be highly correlated, even though they're quite far apart distinctively. Now, if what we're doing with beamforming is subtracting out the activity from all the other parts of the brain, then the activity from this part of the brain that's far away, if it's highly correlated with what's going on in the area we're beamforming in on, then that's going to get subtracted out, and it's not going to be apparent at the spot where we're localized. The saving grace is it turns out that although it formally assumes no correlation, the modeling studies have shown that you can have correlations of like 0.5, which is a pretty strong correlation between two areas, and still be able to differentiate the activity from them. That's for more far apart areas. And when you consider the fact that any activation measure is going to have a lot of noise in it and be influenced by multiple different factors, that's enough that you should still be able to separate out the activities, and results show that. With the sort of extended blob issue, what beamforming tends to do is if you've got an extended blob, it'll just give you something that's kind of in the center of that, and it'll tend to make the blobs look a little more focal and constrained than they would with another, like one of the previous distributed source models that I've talked about. If you've got two sources that are quite close together, it might split the difference and give you a single point in between them, which could be accurate. But again, that's kind of coming from modeling, because in most cases, if you've got two sources that are quite close together in the brain, they're probably part of one active area. There might be exceptions to that, but by and large, these things are considerations in sort of how literally you interpret your data, but they don't fundamentally undermine the efficacy of beamforming. And in other cases, you can get sort of more distributed activities. So this is actually beamforming applied to the same data as in the previous distributed source model result. And again, well, in principle, beamforming should give you more focal kinds of things. In this actual data, it turns out to give you sort of more distributed activity. But if you look at, so intensity here, white is the most intense activation, yellow is sort of the next, orange and red or weaker activation. If you were to threshold this more, you would see that, as stuff drops off, what would be kept would still be the focal part around the superior auditory cortex, which is where we expect it to be. So again, these things, are maybe not the best for determining the true spatial extent of activation, but if you want to figure out where the foci are of activation, they can be effective at that. Again, you do have to take them with a grain of salt and be, cagey about your interpretation, but they definitely add value. And so with MEG, with EEG, the common thing, ERP, is to rely on the sensor-based data and interpret that in terms of things like components or frequency bands, particular time windows, and just average over people based on sensors. With MEG, it's relatively rare that you see an analysis in a paper that's based on sensor data, or they might show that just as sort of step one, but almost inevitably they go to the source localization because, again, it gets you around these issues that the sensors aren't necessarily in the same locations across individuals, and the ability of source localization is quite good compared to EEG. So why wouldn't you? Because you still, and sorry, what's lost here in showing these sort of static blobby brain maps is that this is at one particular point in time. So in this case, we're visualizing it right where the peak is there, just around 100 milliseconds. So with source localization, you can actually do source localization at every time point along. And so this map will dynamically change, and you can actually generate movies from these things where you see how the activation sort of starts and moves through the cortex and so on. And that's something you can't get from fMRI because fMRI doesn't have the temporal resolution. So there's real sort of strength that MEG gives you all the temporal resolution of EEG and higher spatial resolution. And that's why you would pay $500 an hour to do an MEG study rather than simply paying like a couple of dollars per subject to do an EEG study.